waving all the trick-or-treaters and showing up. Talk about defenses. We have both non-specific and specific, all right? The difference between the two is specific defense requires that the cause of the disease, or the cause of whatever's being invaded, is recognized by the immune system. So it has antigens on it, for which our immune cells have receptors that recognize it. So it's a receptor-specific response, right? If that doesn't occur, the body can still defend itself. It's just it doesn't try to decide whether it's self or whether it's not self. It's anything foreign. It doesn't have to be a specific foreign molecule. So some texts will call this component here still the first line of defense. Let me rephrase it. Let's look at. Come back to that one. So they will call the first line of defense mechanical and the second line of defense chemical. And I consider them to be both part of the first line of defense. All right? And the reason for that is because by definition for the first line of defense is any pathogen has not crossed the physical barrier of the body. So there's a physical barrier and a chemical barrier. It doesn't matter if it's being attacked by the chemical barrier, that still hasn't crossed the physical epithelial cells that line the surface, cover the surface of the body, or line the respiratory tract, or the urinary tract, or the digestive tract. So it's kind of the way I describe it to non-major students is imagine battle. The warriors are trying to get on the island, and there's a physical barrier of barbed wire and cement walls and whatever. That mechanical barrier would be our skin, or the lining of our digestive system, or something similar for the reproductive tract. Then, in addition to that, there's hand grenades being thrown. Okay, there's mines that have been planted in the sand. So it's not a physical thing that you have to get over, but it's stuff coming at you that you have to avoid. And so that's what I put in here as well as the first line of defense. Then, once the pathogens get past both the chemicals and the physical barrier, then they're in primarily the loose, irregular connective tissue that's deep to every epithelium, and we have these non-specific responses. And we'll talk about these briefly before we move on to the specific response. Okay? For your first question, lie on there. Roundworms, all right, or filarial nematodes. Nematode is a word for worm, can enter either the lymphatics or the blood vessels. This is the this is the worm that I talked about blocking lymphatics and then causing elephantiasis or enlarged scrotum from lymphatic edema because the fluid is not passing back to the heart. That's been forced across the capillaries. It normally returns to the heart through the lymphatics and the lymph nodes that we talked about. And if it can't get there, then it causes edema. Okay? So these are the worms that cause that process. And what would you expect in addition to the edema that's resulting in the blocked lymphatics, which of these changes in blood cell counts would you also expect? Hi again. So would there be an increase in eosinophils? Would there be a decrease in red blood cells or a decrease in platelets? That's what those terms mean.
What was the first one? A decrease in what?
have sweat glands that secrete a salty fluid. We have the hydrochloric acid in the stomach and the digestive enzymes, and alkaline pH in the small intestine. There's an acidic pH in the vagina, in the tears, in the saliva. We have enzymes that kill off bacteria and antibodies. So the sticky mucus that's present in the respiratory tract and digestive system. So those are all preventing the pathogens from passing between the cells and actually getting into the body tissue, right? The first time. And it's non-specific. So there's no, even with the antibodies, that's a small part. Everything else is a non-specific component of this defense. When we get to the second line of defense, most of the places this is going to occur is in the loose irregular connective tissue immediately deep to the epithelium. That's where we find loose irregular connective tissue primarily. And it's easier for the defense cells to move around within that region and the proteins that are involved. All right, so here's some of the cells that do phagocytosis. So let's even add the lymphocyte to that. It's also phagocytic, although its phagocytosis is primarily involved in the immune system. So I'm going to put it in parentheses because its phagocytosis is not primarily to rid the body of pathogens, but to set up the immune system. But it's one of our phagocytic cells. So we'll look at this process by which we activate lymphocytes, all right? Although in neutrophils, which are our most numerous of our white blood cells and our most numerous of our phagocytic cells, are just involved in physically removing pathogens from the body. All right, then we have natural killer cells. Natural killer cells are confusing because they are a type of a T cell, all right? They are derived from lymphocytes, but they are non-specific. This is what we're talking about. They are a non-specific form of T cells. They do not require, they have receptors, but the receptors, the T cell receptors on T helper cells and T killer cells recognize an antigen, okay? Natural killer cells recognize the constant region of an antibody. So we've talked about antigens and antibodies primarily with blood typing, and we'll finally get to the structure of an antibody later this evening. But antibodies are a form of our specific defense system. And this region right here is specific to the lymphocyte. The lymphocytes make antibodies, so what plasma cells are doing is making antibodies, all right? So all of the antibodies released by one particular B lymphocyte are going to have this region right here that binds to the antigen exactly the same every single time they make them, okay? A different B cell would be making different antibodies, recognizing different antigens. And so this is the part that is specific to our antigen component. This part depends on the class. So we have IgGs, we have IgM and IgA, IgE, IgD. We'll talk about that the last 40 minutes of class. So this part is called the common fragment, or FC. And this region that actually recognizes the variant is called the variable fragment. So we refer to this as the FV site or the FC site. So we only have 5 different types of antibodies, classes of antibodies. And so to recognize that class is not specific. All right, we can have a million different variables all having the same common site. So, backing up a little bit, we talked about mast cells back in Bio 430, and we talked what's the white blood cell that also releases histamine? Anybody remember? One of our 5 types of white blood cells. One breaks it down and one releases it. They're similar in that they both have a horseshoe-shaped nucleus. So the basophil releases histamine. The eosinophil, one of its functions is to break it down, right? So mast cells and basophils. We'll come back here to the natural killer cell in a moment. Mast cells and basophils are both triggered to release histamine when the cell comes in contact. So let's say that we have a toxin, okay, some kind of antigen that you're allergic to that's in your body. And it's recognized by an antibody. And we'll talk about how antibodies can neutralize toxins. But the mast cell or the basophil, and now the natural killer cell, all three of these, mast cell, basophil, and natural killer cell, have receptor molecules that interact with that Fc component. And it's that recognition that triggers them to release the histamine. Okay? So would that be a specific or a non-specific reaction? They're interacting with an antibody. They're interacting with the non-specific part of the antibody. So for the antibody to bind to the toxin here, that's specific, but this interaction here is not. Okay? So this interaction is specific. But this interaction and the subsequent release of histamine is non-specific. Because these three cells don't care, well, that gives them an anthropomorphic mind. They will interact with this Fc constant. That's an IgE. That particular type of the antibody is an IgE. And it doesn't matter what this particular toxin is. That's what we mean by non-specific, okay? So for that particular antibody, yes, it matters what that toxin is, but the mast cell, the basophil, the natural killer cell, they have receptors for the Fc component, and that could be any antigen that's interacting with the variable. And that part right there then is non-specific. So any interaction with the Fc component is non-specific, okay? Because the specificity is at the other end of the antibody. So, coming back now to our natural killer cell, the NK cell. If we have a cell that has an antigen on it for which your body has developed antibodies, so we'll just put some random cell up here that has a foreign antigen on it. And an antibody happens to attach to that and recognize it. We have an Fc component sticking out, okay? So a natural killer cell has likewise receptors that recognize that Fc component. So again, yes, the antibody recognizing the antigen is specific. But the interaction of the NK cell with the Fc region is non-specific. Okay? Now to make it further confusing, that's why it's non-specific, but to make it further confusing, the NK cell's response to this is the same as a T killer cell, which is to send molecules that rupture the cell and cause it to destroy itself from the inside. So the NK cell is going to send molecules that poke holes in the cell and go inside the cell and cause it to break down. So these molecules are identified as perforin. That's the one that perforates the membrane or pokes holes, and granzyme. And And that's what triggers activities inside the cell that cause it to kill itself. So in this activity, if all I were to show you was this response here, it would be the same for both NK and T killer cells, all right? But what triggers that is going to be different. So if you're looking at this and you see the FC component, rather than an antigen sticking out there and interacting with the cell, then you'll know this is an NK and not or a mast cell and not a T killer. So we'll review that when we get to the T killer's response, okay? Now this is a non-specific response. It doesn't require that the natural killer cell recognize anything that's specific only to that particular natural killer cell. All right.
Where
Yeah.
does that FC component come from? Is that just floating around in our body?
The antibodies, yeah. So in other words, a B lymphocyte would have already responded to this particular antigen. This particular antigen would have already interacted with the B lymphocyte that became activated to make plasma cells. And that's where the antibodies come from. So we have antibodies at low levels throughout our body for everything. And maybe there's even been an active response and plasma cells are currently making lots and lots of these antibodies. And so this cell could very well be agglutinated, but it might just bump into a natural killer cell. So both things could happen. Remember I said last night, antibodies never kill anything directly. So this could be another way for an NK cell, for an antibody to be effective, is to activate an NK cell. Okay? All right, fevers are a body's defense. They are non-specific. Anybody remember the part of the brain which is sensitive to temperature? One of its functions is setting the set point. So if your temperature rises too high, above that set point, then you respond by sending more blood to the surface of your body. You turn red, you sweat. If it's too low, blood vessels in your skin and muscles, skeletal muscles constrict, and you might start to shiver depending on how far the temperature goes down. Last night on my way home, it was 53 degrees, I think, 48 to 50, I don't remember exactly what it was when I went home. And I mean, it wasn't freezing, but my fingers were numb by the time I got home. Because in response to temperatures in below 50 degrees, at certain times, not every time, my blood vessels and my fingers constrict, and they go numb, because they don't have enough blood supply for the neurons to work. All right? It's called Raynaud's syndrome, and it's just a sympathetic response to cold. So I can never wear gloves, I have to wear mittens, because if I wear gloves, my fingers get isolated and they just get cold. So that response to changes in temperature is done by what part of the brain? Anybody remember? In 4:30, I know it was a long time ago. You should be high on sugar already. Your brain should be working. Lots of glucose? No? It's going to happen when you get home. Or will it all be gone by the time you get home? So fevers help to maintain the body's non-specific defense by affecting the amount of iron and zinc, okay? And that keeps bacteria from being able to multiply if that is occurring, and increasing the ability of the body to respond to tissue damage to allow increase in repair. So if you probably know, if you have a child, your pediatrician probably told you not to give Advil or Tylenol at the first sign of an elevated temperature. Depending on what your child's natural temperature is, so if it's below 98.6, then you can recognize that 99.5 might be for them the same as 100.5. So you need to be cognizant of your own child's variations. But up to 100.5 to 101, most pediatricians recommend that you just wait, give them fluids, so they don't get dehydrated, but wait to give them medication that lowers the temperature until it's over 100.5 or 100.1, depending on age. And that allows this natural activity, the fever, is a response to reduce the amount of bacteria and viruses. Okay? So, again, it's one of the non-specific activities. All right, inflammatory response itself. Anybody remember what the endocrine type of endocrine hormone that we talked about, reduces the inflammatory response. What are people typically given if they have a disease caused by, or an aggravated condition caused by an increased sensitivity of the body and increased levels of inflammation? You know, back to the very first time when we talked about modified amino acids and glycoproteins and small peptides and eicosanoids and or arachidonic acid derived and steroids. Do you know which one of those is an anti-inflammatory? Steroids, yeah. So people will get them if they have arthritis. Sometimes we'll get steroid injections, all right? And that's because it decreases the cellular activity here. So if you're on steroids for some other condition, and you're going to have surgery, that's typically a no-no because it's going to increase the healing time. It's going to reduce their body's natural response to healing. So typically, if it's possible, the surgeon's going to want your regular doctor to cut back. And that usually has to be done slowly, but cut back on the steroids so that there can be an increased inflammation. So we've talked about, you'll notice there's a gradient of chemicals here, or cytokines. A fancier word is leukokyne, which just means a chemical of a white blood cell. So our mast cells and our basophils releasing histamine are going to increase the permeability of our postcapillary venules. These are starting to tie in together here a little bit. That allows the white blood cells to leave the postcapillary venules by diaphoresis, and they follow a gradient of the chemicals released primarily by the neutrophils that are already there. That leakiness of those blood vessels is typically what causes the redness and swelling at a site of inflammation and infection, okay? And of course, then you have that many more white blood cells that can move to that area. And then they also release chemicals that provide for more movement of white blood cells. So there's an increase in activity of the white blood cells, and then of course for the healing, you've got the fibroblasts. There's a cut that are making the collagen fibers to fill in the holes created by the cut or the damage. But as far as increasing the amount of cells that can, white blood cells that can release their chemicals or do phagocytosis, that's part of the inflammatory reaction triggered by mast cells and basophils. Okay. All of these prostaglandins released there would be considered to be autocrine or paracrine chemicals released right at the site, not having to get into the blood supply to do so. All right, we have two groups of antimicrobial proteins. The previous ones were activities of the body or cells that were involved, and now we have interferons or cells released by molecules released by infected cells, and then we'll look at complement. That will be our capstone for the non-specific defense. So interferons are chemicals released by cells that are already infected with the virus. It's rather altruistic of them. In other words, it helps surrounding cells, but doesn't do anything to help the cell that's making the interferon. Okay? You guys heard of acyclovir? So that's something that is used to reduce the ability of the virus to infect your body, and interferon does the same type of thing. So back in the 70s, interferon was supposed to be the cure for cancer. We give interferon to everybody, it'll block any viruses. Only a few cancers are caused by virus, but it was thought that this is the new thing. Viruses change the DNA and cause cells to become cancerous, and all we have to do is give them interferon, and that will block that. It can help certain cancers and it can help certain conditions, all right? But it hasn't fulfilled its big promise. So what do I mean by altruistic? So this cell is infected by the virus. The viruses take over their host cell by getting the cell to replicate the viral RNA or DNA to produce new viral proteins, which can then produce new viruses, all right? So the cell's actually making the new viruses for them. The cells are replicating, but they're doing so because the infected cell is doing the transcription or the translation of the DNA and both, and making the new viral proteins, which then rupture from the cell and are released into the body to travel to other cells. So in addition to this, when it replicates the RNA, messenger RNA, or goes back to replicate the DNA first, these cells will make these molecules known as interferon. And they are released from the cell with the viruses, and they go into surrounding cells and block the nucleus from being co-opted by it doesn't block the virus from entering the cell, but prevents the virus from co-opting the nuclear material to make the new viral proteins. So it decreases the replication of the virus. Okay? So like I said, it doesn't help the first cell, but it can help the cells around that first cell to ward off the production of more viruses. Not specific. It will happen for any virus. It doesn't have to be just a virus recognized by that cell. All right, then we're going to finish up non-specific by talking about complement. And I don't know if you recall, but I first mentioned complement back in Bio 430 as a form of positive feedback, an example of positive feedback. We have talked about labor, where oxytocin causes the uterus to contract, and that squeezes the fetus, and that squeeze pressure causes more oxytocin to release. We talked about another form of positive feedback with coagulation and the activation of fibrinogen to fibrin. So thinking of a snowball, what event occurs, the thromboplastin activation, or the collagen fibers, the negative charges on a glass wall triggering the cascade of events. So complement is another cascade of proteins. Most of these are produced by the liver, and there are three major events, if you will, or mechanisms of protection. And then we'll look at the pathways. So there is an increase in inflammation, activation of mast cells and basophils by activated complement proteins. There is opsonization. And opsonization is typically illustrated with bacteria. So if we have a bacterium and we coat it with something that's larger than the bacteria, it's smaller than the bacteria but makes the whole thing larger, it's easier for our phagocytic cells to endocytose it. So opsonization can occur with antibodies. In this case, it's occurring with complement. So by coating the outside of the bacterium with complement proteins, it increases the likelihood that macrophages or neutrophils will phagocytose those cells and break them, those bacteria, and break them down. Okay? Then the third mechanism by which activated complement helps to provide protection to the body is the formation of what's known as a membrane attack complex, often described as the MAC attack. So what this is a series of complement proteins, most of which are complement number 9, that end up creating a pore in the cell membrane of the bacterium or some foreign cell that's being attacked, and this allows the contents of the cell to rush out or fluid to rush in and destroy, rupture that cell. Okay? So here's the increase in inflammation. The complement's activating the mast cell. Here is the opsonization, coating of the complement, coating of the bacteria with complement. And here we have the insertion of complement proteins creating this pore. Reminds me of the connexon for the gap junction. It's a little bit larger, okay, because gap junctions don't allow the contents of the cell to rush out. So the cascade that we see up here starts with the activation, and you don't need to learn these in sequence, because like most of our components, they're not activated in the order in which they are necessarily, it's a better order, but not always necessarily the same, the required sequence. So we start out with a complement protein, and in interaction with, it can interact again with an antibody that is coating a bacterium. Does that antibody happen to recognize a foreign antigen? So what was the term that we called this where we coat the bacteria with something? That word over there, that second word over there? Opsonization. So in that case, the bacteria is being opsonized by complement. In this case, the bacterium is opsonized by antibodies. Okay? And this in and of itself will make it easier for a macrophage to ingest it. But if complement proteins recognize this before this happens, then the complement interacts with other complement molecules and splits. Okay? So by coming into contact with this, the complement C3 is going to de-split and form C3A and C3B. Now another way in which the complement C3 can be split or activated is to come in contact with an antigen that's come in contact with complement. Okay? So this would be a complement molecule, and C3 can come in contact with that and also split. So here it's just the antigen all by itself. Here is the antibody. So would this be a specific or a non-specific response here? Where complement is activated because it comes in contact with the antibody. Good, non-specific, because it's coming in contact with the FC part of the molecule, okay? So where complement is activated by an opsonized bacterium, opsonized with antibodies, this sequence is known as the classical pathway. Remember we had intrinsic and extrinsic pathways for coagulation? So where it requires interaction with antibodies is called the classical pathway. Where it comes in contact with the antigen by itself, or even just with the bacterium that has the antigen on its surface, but no antibody, and that still results in activation of the complement, then this is known as the alternate pathway. So this unit has a discussion of two different sets of pathways, intrinsic and extrinsic for hemostasis, for coagulation, and classical and alternate for complement activation. Okay. So the video that I put a link to up on Canvas for you provides a little animation of this. So these are all the different complements over here and the three mechanisms by which they work. And they could have just as well said plasma proteins. Doesn't have to have a clotting. That's what serum is the clotting proteins that take it out. And of course this would be plasma. And you don't have to know specific functions of any of the numbered complements, right? Just be able to recognize the function and what they do. So here we have the antigen. And here this complement C1 is interacting with the FC region of the antibody, not directly with the bacterial wall. So this is the classical pathway. That enzyme now is going to split C3. Notice it was made up of other enzymes that got activated with their interaction. Okay, so that was the classical pathway. And now we can take C3b, and again, you don't have to know the specific complement, and it can act directly with the antigen by itself or the antigen on the bacteria or a virus, whatever it's responding to. It's non-specific. And now it's going to also form C3 convertase and activate more C3A and more C3B. Okay? So the end result of both the alternate pathway and the classical pathway is additional C3B and C3A. And that's why I started that sequence there with C3. So it can interact with the mast cell and cause the release of histamine. That's the increase in inflammation I drew on the board. In itself, C3b can bind to the bacterial cell and opsonize it and make it more likely to be ingested by a macrophage or a neutrophil. And then the next illustration coming up is the back attack. Here's the double layered lipid membrane, and now there's going to be a whole bunch of complement 9 that line up to create this pore, P-O-R-E, and the cell ruptures. Okay. So those are the 3 mechanisms by which this nonspecific protein can increase other nonspecific activities within the cell. It just enhances them and increases their activity, and they're more likely to occur with complement than they would just by themselves. Okay? We have a variety of mechanisms of activating mast cells or getting out histamine. All right, so before we switch to looking at specific immunity, we have one more quiz on this set of slides, and I'll open up the 2 sets. So splitting of complement protein into C3b and C3a as a result of contact with the Fc version. If you're not sure, the answer's on the board. All right. So I don't want you to assume that because you see the word antibody, it's automatically specific. All right. If I mention Fc interaction, it's going to be nonspecific. Which doesn't give me the answer to that, but the answer is up on the board and the previous slide. Okay, any questions on this or what you need to know about it? So recognize those 3 events, recognize the definition of optimization, all right? And that they occur in 2 different pathways by which complement is activated.
Is it me?
All right, so question number 3,
Because it's
and this question came from an exam from last year, previous years. We've talked about this. So a woman underwent a gastric bypass. It wasn't a lap band procedure. 2/3 of her stomach was removed, okay, in order to lose weight. And just a few days ago, she was riding on the bike trail and crashed and tumbled down the bank. The ER team determined that her spleen was badly lacerated and removed it. So she's now at risk for which of the following? So what you want to review is what are the functions of the stomach and the spleen that have to do with the blood and or immune system. Okay?
Do you remember what ridiculous anemia is?
So would blood-borne infections have anything to do with the stomach or the spleen? Or none? Which one? Pardon?
Does work with it.
Which one? Which organ? Sorry?
spleen.
The spleen, okay? And what part of the spleen? What region of the spleen? What do we call the area of cells that help to reduce bloodborne infections?
Is it white pulp?
White pulp, because that's where we have so normally our lymph nodes with lymphatic follicles are filtering lymphatic fluid, and the spleen is the only organ that filters blood. So as it passes through the central arteries or arterioles and then filters out of that, it has to pass by the B cells and the T cells, all right? So if we don't have the spleen, we don't have that ability to filter the blood. Remember I talked about people who don't have their spleen taking preventative antibiotics before dental procedures. So they would be at a risk for infections in the blood. Sometimes streptococcal infections will, the bacteria from the lungs, especially if they've already had a heart attack, will move into the body from the lung tissue. Pernicious anemia, is that going to be in the stomach or the spleen? Stomach. Okay, so what are we missing? What does the stomach produce that she's not getting by having, at least not much, by having 2/3 of her stomach gone? Do you remember the molecule? What is missing in someone who has pernicious anemia? Vitamin B12, and if they don't have their stomach lining, or if they are a vegetarian, not if they're a vegetarian, they're missing the vitamin B12, but if they don't have their stomach, what are they missing that the stomach produces that helps in the absorption of vitamin B12? Remember that one? Intrinsic factor, exactly right. Okay, so by the loss of 2/3 of the stomach, and then what the procedures used to be before the lap band procedure was developed, you still need to have the food pass through the stomach, but essentially it would be clamped and essentially just make it a very narrow tube, all right? So all of this along here would be removed. So the bulk of their stomach would be removed, and just be a narrow tube that would be left. That's a lot of tissue to lose, okay, and a lot of cells making intrinsic factor. All right, what about the fatigue? Stomach or spleen? Both, exactly. So without the intrinsic factor and the pernicious anemia, they're going to have fatigue. And what's the role of the spleen? We're talking about red blood cells and not having enough of them to transport oxygen. So if they don't have their spleen, not the white pulp, but the red pulp, their ability to store additional red blood cells and have them immediately available to the body instead of waiting to produce more. Okay? All right, so just a short little mention of antigens. We'll talk about antibodies more with the B cells, but both T cells and B cells are interacting with antigens. And if you look at that definition, remember I said it was a bit of a circular definition? So an antigen is a substance which stimulates the immune response. The immune response is the response of the body to exposure to antigens, which doesn't explain either one very well, all right? So an antigen has to be something that is recognized by the receptors on B cells and T cells. If it's too big, it won't be recognized, and if it's too small, it won't be recognized. Sometimes we have to add larger molecules to something that we're trying to create an antibody against for science or for medical testing. So when we look at the antigen, this whole thing is an antigen that the body would respond to, but a molecule, protein, a glycoprotein, a glycolipid, but the antibody or the receptor of the T cell is not going to recognize the entire molecule, but it recognizes small specific components of it. And those are called epitopes. Okay? So if we have a protein, say the D antigen on a red blood cell, that's causing antibodies to be made against it, if this is the D antigen, if we look at those antibodies, some of those antibodies would recognize this site, some of the antibodies would recognize this site, some of the antibodies would recognize this site, okay? But they would all bind to the D antigen. So it's just a more specific component actually fitting the receptor of the antibody or the T cell receptor. So If you were taking micro, you've probably heard of polyclonal antibodies and monoclonal antibodies. The clone we're talking about is the B cells that are making the antibodies. So one B cell, all of its antibodies are going to be to one particular epitope. To get antibodies made against this antigens of various epitopes is going to take a different B cell for each epitope. Okay? So if we have polyclonal antibodies, they're all for this antigen, but they are to different epitopes and they are made by different B cells. The monoclonal antibodies make a test more specific. You have fewer false positives or false negatives if you're using monoclonal antibodies. That's why they came into interest. So just as an aside on the procedure, let's say that we have placed the antigen in an animal like a mouse. Sometimes they're too small and you have to add an extra molecule to it to make the mouse sensitive. And the organ that's usually harvested, removed from the mouse for this is the spleen. You can imagine that there's thousands of B lymphocytes in that spleen that need to be separated so the particular antibody that's needed can be isolated, usually on a column. So to simplify things, let's say we have 1,000 cells, 1,000 lymphocytes, B lymphocytes, in 1 ml of growth serum. And we want to be able to separate them out into individual cells so that we can harvest enough antibodies to determine which one we want. So the procedure that's used to do that is to add 999 mils of serum, growth serum. So now we have 1,000 mils, it's just a liter, it's not too big to handle. If it's a higher concentration, we can just take a microliter, or 5 mils, or 1 mil, various amounts. And then we have a growth chamber of 1 mil squares. And we pipette 1 mil of this solution, because on average there would be 1 cell now, instead of 1,000 cells per mil, there would be 1 cell per mil of growth fluid. So if we pipette a mil into each of these growth plates, chambers, there will be some that have 1, there will be some that don't have many, there might be some that have 2, but on average, most of them are going to have 1 cell. Okay? That cell is going to undergo mitosis. And again, and again, and again. And each one is a daughter cell of the original parent. So just like all the cells in your body have the same DNA, all of these cells in this mill are going to have the same DNA. They're going to make the same antibody, and then that particular mill, they would be monoclonal. All right, so that's kind of how they, and then they would run tests on them to decide which antigen. The way this is done is if you know the antigen that you're trying to, and the epitope that you're trying to specify, you make a column of beads, typically glass or plastic beads, and chemically attach the antigen to them. And then you run this solution of the antibodies of the cell, and well, you isolate the antibodies through that, and all if they were the right antibodies, they would bind to that antigen, and all the rest of the antibodies would flow through. But now you need to get them off of that bead, and so you change the pH, or you change the chemistry of the solution, and they now let go of the bead, or that entire column. It's just a way of isolating the cells or proteins from that. Okay, so I just want you to understand monoclonal and polyclonal and epitope versus antigen. Okay? All right, so back to looking at cells. We have T cells, remember from Monday, Tuesday night, we have T cells, and we have B cells, and we have the macrophages, and we have the reticular cells for our lymphatic system. And looking at the actual immune response, we no longer need the reticular cells. Okay, they don't have anything to do with the immune response. The reticular cells are only involved in the supportive structure of the organs. So let's look a little bit more closely at these cells. And T lymphocytes, we're not going to consider the NK cells here because they're not involved in specific defense. T lymphocytes can occur in three major forms as a T helper cell. And both B cells and T cells will not activate to their function unless they are aided by chemicals from the T helper cell. Okay, so the T helper cell is involved with both the T cells and the B cells. Then we have the T killer cells, also known as cytotoxic. They have a direct cell-to-cell interaction with anything they're going to kill, all right? And as I'll illustrate to you later this evening, they are involved primarily in transplant rejections, in fighting tumor cells, cancer cells, and virus-infected cells. Okay? Not fighting viruses necessarily, antibodies can do that, but fighting the cells that have been infected by the virus. Your own cells that have been affected by the virus. And then T suppressor cells do just what they say, they tap down the immune response. Remember we talked about in the placenta, when it implants, how in the decidua around the chorion, that there are fewer numbers of white blood cells, and there's more of these T suppressor cells, so that the maternal tissue accepts. Remember we said half of the fetus's cells, DNA protein came from the dad, and that could be very foreign to the mother. In fact, it could cause some women to reject any implantation. So T suppressor cells are involved in suppressing an immune response. So T lymphocytes are found in all of the following locations except one answer. May I erase the board? You guys are thinking about that? We kind of had this question Tuesday night with the histology. Okay? One more time on number 4. Okay. All right, so here are the T cells, and then we have the B cells. And you can see when the B cell is not activated, it's very difficult to tell the difference between the B cell and a T cell. One thing is, if shown, the T cells are going to have this direct cell-to-cell connection when they are actively fighting. So a tumor cell, a virus-infected cell, a transplant cell, the T cell is going to bind to an antigen on that foreign cell and send out perforin and granzyme, like we talked about with the NK cell. But when it's isolated by itself, and the plasma cell has not been, the B lymphocyte has not been activated to a plasma cell, then you can't tell. Okay, they're just lymphocytes. So here's the appearance of the plasma cell, and it's unusual to actually have it with red blood cells around it, because this activation usually occurs in the connective tissue. Okay? And then I have listed macrophages and dendritic cells, which are derived from the same precursor to the monocyte. So here's a macrophage, up here's a macrophage, okay, although they usually are also in the connective tissue, not with red blood cells, or white blood cells. But dendritic cells are found in the stratum spinosum of our skin. So they're very close to any site that might have an invasion. And we call these cells, as you can see in the smaller print, an antigen-presenting cell. Okay, we're going to look at that later today. So we have 3 major cell types that are considered to be antigen-presenting cells. And this is what is required to activate a T helper cell. Okay? So we have APC, means antigen presenting cell. So we have macrophages, macrophages, dendritic cell, which is a more star-like form of a macrophage, okay? And we have B lymphocytes. To be an antigen-presenting cell, it has to be phagocytic. All right, so all three of these cells are phagocytic. But notice I didn't put neutrophil up there. A neutrophil and an eosinophil are phagocytic as well, but they are not antigen-presenting cells. They break down their antigen, they don't put it out on its surface in order to activate the T helper cell. Okay? So here's a general overall diagram. Here's our antigen-presenting cell, internalizing the antigen, the bacterium or the virus, via endocytosis, phagocytosis. It is broken down by lysosomal enzymes. And then in a process that we'll look at shortly, those enzymes are, those proteins have been broken down, are presented on the surface of that antigen-presenting cell. That's how it gets its name. If the T helper cell's receptor recognizes that antigen, this is a specific reaction, by the way, right here. All right, so it's the T cell receptor that's recognizing the antigen, not an Fc component, but the actual antigen. Then there are chemical reactions between the macrophage and the helper cell that turn it on. And now this active T helper cell can activate either T killer cells or B cells. If it's the T killer cell, then it becomes sensitive to that same antigen and has that cell-to-cell reaction and kills it with granzyme and perforin. If it's a T helper cell, when it finds the antigen in the body, then it makes antibodies, and those can spread throughout the body. Okay, it doesn't have to be a cell-to-cell interaction for that. All right, so let's look at what we mean by that molecule that we saw here that was used to present the antigen, okay? These molecules have a couple of different names. Major histocompatibility complex, or they're also known as human leukocyte antigens, so HLA. We're going to go with the major histocompatibility complex, all right? So we're going to look at class 1 and class 2, major histocompatibility complexes. And we'll do class 1, and then we'll take a break and come back and do class 2, okay? And then we're done with T cells. We'll have a break so you can study for the quiz. And then we'll come back and finish up with B lymphocytes. So we're going to look at MHC1 first, which is major. Histo refers to tissue. Histocompatibility. And complex. Okay? So these are formed on chromosome 6. Maybe I can get that to show it better if I give it a white background. Actually, before I do that, let me change this to black. And I'll get the alignment again. Get my whole slide in. So the chromosome 6 has on its region various different genes for making HLA, which is the same as our MHC. Okay? And so MHC is a complex of antigen-presenting proteins that are found on every cell that has a nucleus. So this is why red blood cells are not, they're not considered to be, you don't have to take anti-rejection drugs to get a transfusion. okay? Because red blood cells don't have a nucleus once they leave the bone marrow, and therefore they don't have cell markers that mark them as your cell. So they're not recognized as foreign self when they're given transplanted to somebody else. So they work okay on that. So every cell that has a nucleus, including our antigen-presenting cells, are going to have MHC class 1. So remember when we talked about the thymus Tuesday night, and I said it was a nursery for developing T cells? Good. So the young T cells migrate to the thymus, and we want them to be able to recognize the MHC class 1 molecule. Because if they don't interact with the MHC class 1 molecule, they're going to be of no use in reacting to a foreign transplant cell that has a MHC class 1 molecule on it. So it has to be able to respond to MHC class 1 molecules. Okay? Apoptosis, by the way, means cell suicide. The cell kills itself. So then it passes that test. But then it's tested to whether or not it reacts to all the MHC1 antigens that we have in our body. All right, so various cells are passed through the thymus, and we want it to not react strongly. It's okay if it reacts a little bit of that, but if it reacts strongly, then it's going to cause autoimmune diseases in your cells. And we don't want that to happen. So those cells are triggered to be killed by the cells in the thymus. So once they leave the thymus, all right, and take up residence in the body, they're capable of reproducing and maintaining a T cell population in your body for the rest of your life. T helper cells, by the way, are the ones that are invaded by the HIV virus. And so once they kill most of those T helper cells, both your T cell and your B cell system no longer works. And that's why it's so devastating. We don't have any of that immune system left. So let's look at the process by which the MHC class 1 molecules work. And they're made from the genes on the chromosome 6. And so messenger RNA is formed and translated by ribosomes, just like we learned about in protein synthesis. And class 1, MHC class 1 molecules are inserted, they're not just found free, they're inserted into the membrane of the endoplasmic reticulum. Okay? So let's get to that point first. So this is any nucleated cell here that I'm illustrating. We're inside it, we're not looking at the outer membrane. Here would be a endoplasmic reticulum. And the synthesized class 1, MHC class 1 molecule, after it's made via the ribosome and messenger RNA, instead of being sent to the Golgi, is inserted into the endoplasmic reticulum. Okay? Well, how do we get our antigens in there? Other proteins are made by the cell, right? Based on the genes on that cell, it's going to make other proteins. And so those proteins are found either already in the ER, because they're proteins that are going to be sent out to the cell, or they are proteins that are going to be used for enzymes in processes utilized by the cell. So they're just made by free ribosomes. Okay? So we call these proteins endogenous. Meaning made within. So they're a little bit too big to pass through the endoplasmic reticulum. So there are enzymes available in the cell that break these down into functional pieces. Some of them are going to work in the cell, but some of them are going to be broken down. And there are special transport antigen proteins that are in the endoplasmic reticulum that will allow these pieces to enter into the endoplasmic reticulum. Okay? Once place they will associate with the MHC class 1. This is not specific here, okay? So the MHC class 1 molecule will bind to any molecule that fits it, as far as size is concerned, that's in the endoplasmic reticulum, okay? So different proteins will go, antigen size molecules will go to different MHC class 1 molecules. All right, then this pinches off, and it goes to the cell surface. So we have a sac containing our MHC class 1 molecule, containing the antigen. And then that's going to insert, the membrane's going to bind with the outer cell membrane. And because of the way it's situated on that, it's now facing outside the cell. Okay? So there's the antigen on the cell surface of an MHC class 1 molecule. So endogenous proteins. Okay? Or they could be glycolipids or other molecules made by the cell. But they're usually a protein attached to a lipid or a protein attached to a carbohydrate. Now, why does this work with viruses? Why would a viral protein be attached to an MHC class 1 molecule? Remember how we said viruses work? They co-opt the host cell's nucleus and get the host cell's protein synthesizing apparatus to make more DNA, or to take the RNA that's in the virus already and transcribe that to make viral proteins. So those viral proteins are going to be in the cytoplasm, and the cell doesn't know that the nucleus wasn't supposed to make them. And they're going to be treated just like any other endogenous protein. And they're going to be inserted through the TAP into that cell's MHC class 1 molecule. Because the cell can't tell the difference, whether it made a viral protein or it made a protein from its own nuclear genetic material. Okay? So that's why viral proteins can be associated on the cell surface with an MHC class 1 molecule. All right, what about transplant tissue? That one's a little easier to see, because my heart tissue has my MHC class 1 molecule with my self-antigens on it, okay? So if my heart needs to be given to somebody else, it's going to be different from their self-antigens. But it would be this normal process by which my cells put it on the cell surface, which is that other person's going to recognize it as foreign. What about a tumor cell? It's still my cell, right? So if I have a squamous carcinoma and I have tumor cells, how did those cells get foreign self-antigens? Just being a cancer cell means the DNA has gotten messed up. And that means it's making proteins that would have been exposed to the T cells in my thymus. So the T cells are going to recognize those new proteins made by the modified DNA of the cancer tumor cell as foreign. But it's still going to be my cell making them. So it's still going to end up in the ER and on MHC class 1 molecules. Okay? So, as I said, this is how we mark ourselves as belonging to us. Normally, an MHC class 1 molecule would have your self markers, what we mean by self-antigens. All right? And if it's co-opted by another cell, or it's a foreign organ to your body, or it's a modified cancer cell that's making foreign proteins, that's how we get non-typical self-antigens on the surface. All righty? Okay, actually let me do the MHC2, and there'll be a better break before we take a break. So MHC1, any nucleated cell, endogenous proteins. We'll come back to the CD4 and the CD8. All right, MH class 2. So let me grab my eraser here, then if you need to take a picture of this, it's just a simplified version of what you have on the PowerPoint. MHC class 2 are found only on our antigen-presenting cells. So it's found only on the macrophage, dendritic cells, or B lymphocytes. But remember, those cells all have nuclei, so they're all going to have the MHC class 1 as well. Okay? It's not an either or, as far as they're concerned. So here you can see the source of the, sorry, didn't mean to walk across your picture. The source of the antigen now is from outside the antigen presenting cell. We're going to use a macrophage here, all right, to process this. So it's called an exogenous peptide because it was formed by another cell and is now being internalized through phagocytosis. So it is exogenous. So instead of any nucleated cell, we're going to have antigen presenting cells, okay? And instead of endogenous peptides, this is going to be exogenous. So our MHC class 2 protein is still made by the endoplasmic reticulum, okay? So we'll draw the endoplasmic reticulum first. And I'm going to put both an MHC1 and an MHC2 on there just to remind you that these cells have nuclei, so they have MHC2 molecules as well. I mean, MHC1 molecules as well. And here is the MHC2 molecule. Okay? And this is our ribosomal ER. All right. Now this gets pinched off. And so we have a vesicle that has in it MHC class 2 proteins, but it's now separated from the ER. Which means that our exogenous peptide doesn't need to move into the ER in order to associate with that MHC class 2 molecule. So how do we get it? Exogenous. So we have a cell membrane undergoing endocytosis, and we'll keep using a virus. And instead of co-opting this cell's nucleus, which you can do, all right, but in this example it's not. So instead of co-opting this cell's nucleus, this is going to be endocytosed, creating its own little vesicle with a virus, probably thousands of viruses. And to break down the viral protein, this is going to fuse, because the cell doesn't know it's a virus, it thinks it might be food. So it's going to fuse with the lysosome. And the protease enzymes, which function to break down proteins, are going to break down that virus into a whole bunch of different proteins. Okay? Some came from inside the virus, some were the viral coat proteins. They're proteins, and so the cell breaks them down. Now, this vesicle is going to fuse with the lysosomally active one. So I'll draw that down here. There's a snowman. And now we have our MHC2 molecules. That now have access to the viral proteins. Same mechanism. This will pinch off. And head towards the surface membrane I'm the antigen presenting cell, now presents the antigen. Okay? So two different mechanisms for getting a protein, or parts of a protein, antigenic smaller parts of a protein, onto the surface. So the MHC class 1 is how we have our cell markers on all of our cells that have nuclei. And they're normal for us. Our bodies should not react to them unless it's been modified by cancerous gene changes, all right? Or viral proteins because that cell was infected, or it's tissue from another person. So we're going to look at how the cell uses MHC class 1 molecules to activate T killer cells, and MHC class 2 to activate helper cells and B lymphocytes. Okay, so that's what we'll do when we come back. We'll finish up with T cells, then we'll take a lab time to get the lab quiz out of the way, kind of break up the lecture a little bit, and we'll come back and finish up with B cells and antibodies. Okay, so why don't you come back about 20 minutes.